There we go. So I will talk a bit about what is differential expression. I should also mention that most of these slides, it was actually Olga at NBIS that was supposed to have this lecture, but she had some problems with disease in the family, so she couldn't work on it. So I started last week to look at this lecture. So most of the material is hers. I will talk about what is differential expression analysis, some common methods, a little bit on performance, and then some things that you need to think about when you do differential expression. So what is differential expression? Basically, if we have single cell data, we define, in this case, 2 clusters. And then we want to get a list of genes, right? Genes that are up or down regulated in either of the clusters. Normally we get a fold change between them and a p-value that tells us how significant this difference is. And quite often you can see by eye if you believe it to be significant, like in this example. I'm pretty sure you can say yourself which ones are up and down regulated. Or not really significant, like here. This one looks like it's upregulated in cluster 1 and this one downregulated. And the question is really to decide when is this difference significant? And we can use a lot of different tests to do this, of course. The other issue is that in single cell data, it quite often looks like this. Would you say that this is a significant change or not? If you just count the proportion of cells in the 2 clusters that express the gene, it might actually be a significant change, even though the level of expression is the same in both cells. And this is quite often the case, that it's more like the frequency of detecting the gene in one cell population versus the other. That tells us something rather than the level. And especially just looking at the means when you have high level of zeros for a gene is very meaningless, basically. And here is just a citation from a paper that a lot of methods still think about single up and down, while not really taking the amount of zeros into account. So imagine then we have our 4 clusters or 10 clusters, whatever we had from our data set, and we want to find genes that define each cluster. We can do pairwise, each cluster versus each other cluster, which ends up then with, in this case, quite a lot of different gene lists to go through and compare. We can take one cluster versus the rest. So basically cluster 1 versus the other 3, cluster 2 versus the other 3, and so on. And there's a lot of ways of doing it. The most common way to start with is to just do this 1 versus all for each cluster to get a list of genes for that cluster. And you can also specify, if you just want like typical markers, you can take only upregulated genes per cluster. So it's easier to interpret the results to start with. I want to find markers for cluster 1,